Okay, class. So we just discussed about the components, right? What are the components and what are the main features in those components? So now we'll be discussing about the affinity. Okay. So for example, you see now there is this tRNA, RNA, small subunit and large subunit. So how do they react with each other? Okay. So you see, for translation to happen, which means growing of peptide, we need amino acid, right? We need amino acid. We need amino acid right over here. You can see we need amino acid. So this amino acid will react with tRNA because tRNA is the one which carries the amino acid, right? So in the very first, what happens is that the tRNA, which has OH group in its 3' end, will react with the amino acid. But these amino acids are not reactive. You need to make this amino acid reactive. That means they need to be activated. So therefore, this amino acid will be first activated by the help of ATP. Okay? And once these amino acids are activated, now this amino acid can be loaded to the OH. So this this is called activation of amino acid, which is same in both prokaryotes and eukaryotes. And after once these are activated, this will be loaded onto your tRNA, which is called charging of tRNA. Okay, so this is also similar in case of prokaryotes and eukaryotes. But what we have to understand is that the amino acid, the very first amino acid, is methionine in case of prokaryotes as well as in eukaryotes, except that this methionine in case of prokaryotes needs to be formilated. Okay, and this formylation of methionine takes place or is catalyzed by tetrahydrofolate transferase. Okay, so formyl is basically one carbon, right? And one carbon compound is transferred by tetrahydrofolate transferase. What is the source of tetrahydrofolate transferase? Basically, tetrahydrofolates are the activated form of folic acid, right? And these are therefore obtained from folic acid, which is vitamin B9. So in this way, you see amino acid is activated, but formalin group is added, and once these activated amino acids are loaded, this is called charging of tRNA. It happens in both the cases. Now this charged tRNA can either react with the mRNA or with the small subunit of ribosome. How? Because you see this tRNA has anti-start codon and we have a start codon in the mRNA, right? So these anti-start codon can actually base pair with this start codon. But if tRNA comes over and base pairs, this will not be stable, right? That means it has affinity, but it won't be stable. Therefore, instead of tRNA first going to mRNA, will go to your small subunit. Why? Because small subunit has a P slot or P site. Okay, and this tRNA can go and sit over P site. But what about A site and E site? What happens is that E site is blocked initially, and E site is blocked initially by initiation factor 1 and 3, and your A site is blocked by initiation factor 2. So that means E slot is only available. Therefore, the first tRNA can formalinated methionine will come and sit over here in the P site. Okay. Now, since this tRNA along with 2-GTP sits over here, this is called pre-initiation complex. The same thing happens over here in case of eukaryote. Okay. Now, once your tRNA is seated over here, what happens is that now, because this tRNA has anti-start codon as well as this small subunit has anti-Santalgado sequence, right? So both of these have affinity for this and this. It is same in case of eukaryotes, right? This loaded tRNA will go and sit here, and because of the anti-start codon in tRNA, which can base pair with the start codon as well as the anti-Kojak consensus sequence can base pair with the Kojak consensus sequence in the mRNA. Therefore, the mRNA will rush over to this small subunit where they can base pair, and Kojak consensus sequence as well as Sandalwood sequence are in such a way that as this anti-Koja-consonance sequence or anti-Sandalwood sequence base pairs with their respective Sandalwood sequence and Koja-consonance sequence, the P side will exactly fall upon start codon. Okay, the P side will exactly fall upon start codon. That means your tRNA which is in the P side will base pair. Okay, so this is the affinity. And once this is formed, your large subunit initially won't cap your small subunit. That's why I have removed this part. Why? Because like I said, the initiation factor 1 and 3, actually 1 and 3 in case of prokaryotes as well as in case of 1a and 3 initiation factor in eukaryotes are inhibiting this large subunit to come and cap your small subunit. Okay, that is why your initiation factor 3 is also called anti-association factor. Here also you can see the 3 factors called anti-association factor. Why? Because these are repelling. These are repelling or inhibiting the large subunit to come and cap over here. That is why. Okay. So what happens is that is why your large subunit comes at the end once your tRNA mRNA has combined with your this small subunit. Okay, and once everything is ready, then what happens, both of these are removed. Okay, both of these are removed, and as these are removed, now simply your large subunit will come and cap. Okay, as these are removed, this will come and cap. So now you have 70s ribosome, you have 80s ribosome. Okay, so let's again revise the affinity. Amino acid has affinity for tRNA, tRNA has affinity for B side and P side with the small subunit, and then the mRNA has affinity with the small subunit. Once these are done and the initiation factors like 1 and 3 are removed, then your large subunit will come because this large subunit has affinity with the small subunit, and then that is how your whole initiation complex is formed. Okay, and later on elongation takes place and then finally termination. So in the upcoming videos I'll be talking in detail about how the initiation takes place in translation and how elongation and termination takes place.